Hello. We are now a couple minutes past the hour, and I'm absolutely excited to begin. So why don't we jump in to this exciting edition of MIT Abstracts. I would like to give a warm welcome to everyone who's logged on so far from all around the world. Welcome to Nord Anglia students and their teachers, and welcome to everyone on this really beautiful day. And I'm excited to share with you all an absolutely stunning talk today. I'm super excited for it, and I hope that you guys are just as inspired at the end of it as I am already. I am your host, Fatima Husain, and I am a PhD student here at MIT studying ancient Earth and how microbes have evolved throughout Earth's history. I am very excited to be here and to host this series and bring all of these exciting people from MIT straight to your screens, regardless of where you are around the world. So I wanted to cue some folks into our series and its name. And the abstract, as you can see, it's just a few sentences describing what that paper is about and what it means. And, you know, I think it's a perfect name for our series, MIT Abstracts, because what we're doing is we're bringing abstracts of exciting, inspiring folks at MIT straight to you through this series. So you're going to get these inspiring snapshots of people. So without further ado, I'd like to introduce our exciting guest, Jordan Harrod. I'm a big fan of her work myself. Jordan is a systems neuroscientist, and she's a medical engineering and physics PhD student, and she's a part of the Harvard MIT Health Sciences and Technology program.
I'm super excited to be here and to be telling you guys a little bit about how I made my journey through science, which, as you can see from the title, I would say is nonlinear. And I'll get a little bit into what that means a little later. But first, to introduce myself, hi, I'm Jordan. As Fatima mentioned, I'm a PhD student in the medical engineering and medical physics program through the Harvard MIT Health Sciences and Technology program, which is a bit of a mouthful, but essentially what that means is that I'm doing a PhD in applying engineering to different types of medical challenges. And my program lets me take classes with med students, as well as taking basic engineering classes so that we can really understand how different issues in a hospital affect our research in the lab. I'm also a YouTuber, so I have a YouTube channel on the ways that artificial intelligence and algorithms impact our lives every day. It was something that I started as a hobby when I started grad school, and it's blossomed into this great community that I love engaging with every week. I went to Cornell for my undergrad, which I'll talk a little bit about my journey in another couple slides, where I studied biomedical engineering. So I've always kind of been interested in the intersection of engineering and medicine. And I've also always been into sports, as you can see. So my fun fact is usually that I've played most individual sports competitively at one time or another. I was definitely not a stereotypical nerd in high school. I was a 8-season varsity athlete. And when I got to college was when I started diving more into the science side of things, but I'm still very much into fitness and sports. And yeah, I'm excited to talk to you guys about how I got here and what I do now. So let's start with what I do now, which is a PhD. And a lot of people don't necessarily know what PhDs are when they're in elementary, middle, or high school. I didn't necessarily know what a PhD really was until probably I started, but I didn't know what PhD students do until I started getting involved in research. I was really lucky to get opportunities to do research in high school and learn that research isn't just doing a lot of Googling on a topic and then writing a paper, which is what I thought research was for a really long time, but it's getting to do experiments and tinker with different systems and try to understand things about science and life, in my case, that we don't necessarily have a great handle on right now. So for a PhD, I do experiments. I write up papers on the experiments that I do. So there was an example of one of the papers that I and some of the grad students that I worked with as an undergrad wrote a few years ago. you work a lot with other people. So I think collaboration is huge in doing a PhD. There's definitely no way that I could get the stuff that I do done without working with other people. And generally just learning a lot and discovering new things that humans didn't know before. It's also getting paid, which a lot of people don't necessarily know about. So unlike when you go to college, where you have to pay the school to learn, in at least science and engineering PhDs, you typically get paid to learn. So if it's something that you've been interested in or considering, but the financial aspect was a bit of a concern for you, just know that there are options for you to do school as a job, which is basically what a PhD is. So when I was your age, I thought that people kind of knew what they wanted to be in high school and that you couldn't really change your mind after that. And whatever path you were going to go on was the path that you decided when you were 17. And at the time, I wanted to be a doctor. So clearly I'm not a doctor now, and I have changed my mind. And in fact, I've actually changed my mind a lot. So I went through a ton of different phases in terms of what I wanted to be. Starting off with a doctor, I was worried that I was going to hurt my patients accidentally. So then I decided that I would be a mortician because you can't hurt dead people. Then I was thinking that I might be a teacher because I had gotten into classroom education. I happened to work with a program at Cornell that's called Splash. They also have it at MIT, which I did in high school. And it's a school program where you get to teach a course on any topic that interests you, basically. And I found that I really liked teaching kids about how to engineer superheroes for a couple hours a weekend by going over basic things in biomedical engineering and then giving them different like X-Men superheroes and being like, how would you actually engineer these powers into a person and like, what would that look like? After that, about midway through undergrad, I was thinking that maybe I would go back to being a doctor. I started undergrad as a pre-med student, so taking courses to prepare me for medical school. I was leaning towards being an MD PhD, so doing like a lot of school, about 8 or 9 years of extra school after college, where I would get both my PhD in an engineering field and then also go to med school and do some combination of practicing in a hospital and doing research in a lab. And at some point in college, I figured out that at the end of the day, I didn't really want to be a doctor. I wasn't necessarily interested in treating patients, but I was still interested in tinkering with medicine and medical systems and human biology. So then I thought I would be a biomedical engineer. I actually got to work at Stanford, which I'll talk about a bit in the next slide, and got to work with machine learning systems. So I thought that I might be a computer scientist who works at the interface of medicine. And now I'm a PhD student. And so I focus currently on neural engineering and making YouTube videos, which was something that definitely came up kind of at the last minute right before I started grad school. So this is all to say that you can always change your mind. You don't necessarily have to know exactly what you want to do when you do it. And there's a lot of flexibility, even if it feels like you're locked into a certain career path. And so importantly, I wanted to note that even with this winding path, I don't know what I want to do after this necessarily. I have thoughts. I think that for what I do, which we'll talk about in the next couple slides, there aren't a ton of jobs that let you do that. And so you don't necessarily have to know what you're going to do when you start your PhD. You don't necessarily have to know what you're going to do after you're done with college when you go into college. But my piece of advice for that would be to, if you can, find opportunities to try the different things that you're interested in. Because for me, figuring out that I wanted to do a PhD in the type of research that I do came because I tried a bunch of stuff that I thought that I was interested in and realized that I either was really interested in it or it wasn't the thing that I thought it was. So I thought that when I went into college, I was going to go into pharma. I was going to work at a drug company. And then I spent summer at Novartis and figured out that maybe that wasn't the right place for me. So if you can, I would highly recommend finding opportunities to try out the different things that you're interested in, learn more about what it's like to do those things, both in terms of what your long-term goals for those projects are, but also what it's like to do them on a day-to-day basis, because even if the outcome seems really, really cool, you might not actually like what people who reach that outcome have to do on a day-to-day basis. I'll also note that I definitely didn't do this alone. I had tons of mentors, from my friends to my family to my professors. And I think that I definitely wouldn't be here if it weren't for them. I had a ton of support from people who, made time for me, answered my questions, connected me to those opportunities that I might not have otherwise. And so if one thing that you take away from this, I would say it's to find mentors that will support and empower you, because it will make a huge difference, both in terms of the opportunities that you get access to, but also just in terms of the amount of work and stress that you'll be under, because you're not going to be doing it alone. You're going to have people who can support you the whole way there. Oh, I forgot that was a GIF. So what do I do now? Not this, actually. So this is a video from one of my labs. It's a method called Brainbow. And it's essentially this thing where we can take a piece of tissue from the brain and take enough pictures of it to make a 3D version that looks like this. To make it easier to see all the different parts of the brain, we also stain different things with different colors. So the brain does not normally look like this, but it's a really nice way of looking at the brain in this really colorful and kind of beautiful way. And it's systems like this that I was interested in developing when I got into grad school. As I mentioned earlier, what I actually do could be best described as systems or computational neuroscience, which is modeling the brain using code, trying to understand how different parts of the brain interact with each other and how we can change how certain parts of the brain function in order to change how other parts of the brain function as well. So when you're in grad school, you work in a lab with a professor who's usually your advisor. I happen to have 2 advisors, which is a bit unusual, but I think it was the best fit for me. My first lab is called the Neuroscience Statistics Research Lab, which is led by Dr. Emery Brown, who's on the top right. And in that lab, we try to basically model different systems in the brain using statistics and machine learning. One of the things that we focus a lot on is general anesthesia, which is a combination of medications that put you to sleep and make it so you don't feel pain when you get surgery. And in fact, Dr. Brown is an anesthesiologist or a doctor who oversees anesthesia and surgery. And so at one point, there was someone in my program who had appendicitis and had to get their appendix removed. And as they were being wheeled into the OR, they looked up and my advisor was the one who was managing their anesthesia. So he works in one of the hospitals around here and is usually during non-COVID times in the clinic about 1 or 2 days a week and practicing anesthesia, which is pretty cool. So we get a lot of clinical information in terms of, for the systems we're designing, does it work in our lab? Great, but does it actually work in hospitals? Like, can doctors use this? Does this make sense to the people who would be using it? Which is really valuable for the type of work that we do. My other lab is the synthetic neurobiology group, which is led by Dr. Ed Boyden, who's on the bottom left. And our lab focuses on developing new tools and methods to help us learn more about the brain. So our lab motto is watch, perturb, and map, which basically means that we develop different tools to look at different systems in the brain or sections of the brain to change the way that the brain functions, and then to better understand how and where different parts of the brain connect to each other. And I'll say here that my PhD program lets you do something called rotations, which essentially means that you don't have to know exactly what you want to do when you get here, and you don't have to have someone that you're already planning to work with. You can try out a bunch of different labs and see which one's the best fit for you in terms of your research interests, in terms of the lab culture, in terms of the mentorship that you would get from that advisor. That was definitely preferred for me as someone who didn't necessarily know exactly what I wanted to do when I started my PhD. And I think it's a great way of figuring out what you want to do and what it's like kind of on a day-to-day level in different labs and for different kinds of science. So personally, for my PhD work, I'm interested in anesthesia, non-invasive brain stimulation, and machine learning. Specifically, I'm interested in what happens to the brain when you're taking anesthetic drugs and whether or not we can replicate those methods using other types of tools or other types of drugs. So for example, when you get a paper cut, there are cells in your finger that sense that pain, and they send that information to your brain, and your brain knows that you have a paper cut on your finger. However, when you're under anesthesia, the drugs prevent those cells from telling your brain that it's sensing pain by activating or deactivating certain regions of your brain. In theory, there should be other ways of doing that than just using drugs, things like electrical stimulation. Mostly electrical stimulation is what we're looking at. And so I'm interested in finding out what those methods are and what those areas of the brain are and how you activate and deactivate them in a way that gives you the same effect as anesthesia, but without the drugs. So one potential way of doing that is called temporal interference, which is the picture that you see here. It's essentially a way of using electric fields, which are the lines that kind of go in a circle between the squares that are on the person's head, to activate neurons that are deep in your brain, which happens to be where we think anesthetic drugs work. And our idea is that unlike a lot of the methods that we normally use in research, which involve like implanting electrodes in people's brains or really in animals' brains in order to do this kind of stimulation, that doesn't really make a ton of sense for someone who's coming into the hospital to have surgery. Like if you're trying to get your appendix removed and we have to install electrodes in your brain first, then that doesn't really help you. We're interested in looking at methods where we can just place things either on or near your head, and it can do the same thing as those electrodes that would be implanted deep in your brain. So the other thing that I'm interested in is machine learning, and I'm specifically interested in whether algorithms can learn how to control anesthesia. This is something that we think about a lot in the neuroscience statistics research lab, and there are a lot of people in my lab who work on these types of problems. So some examples of machine learning for people who aren't familiar would be things like self-driving cars, or when YouTube recommends different videos for you, or when you start typing something into Google and it like provides recommendations on what you think you might be searching. And in essence, it's designing algorithms that can learn patterns from data and then applying those algorithms to new data that ideally has some of the same patterns. So we're interested in seeing whether or not you can give machine learning models data on here's the amount of anesthetic drug that we were giving X person, here's what their brain waves looked like when we gave it to them, and should we increase the dosage, decrease the dosage, keep it the same to make sure that they stay at a certain level of anesthesia throughout the whole procedure, as well as to make sure that they go under in the first place and wake up at the end. In short, my PI always says that I'm trying to put him out of a job with my PhD thesis, which is somewhat accurate if it's successful, but I think that that's probably not going to happen by the end of my PhD, but we'll see. So of course, being a PhD student is not all that I do. In fact, for most PhD students, I would say that we all generally have lives outside of lab. One of the big things that I do outside of lab is run a YouTube channel on artificial intelligence and algorithms. So I make videos on everything from whether or not the AI system that's being used to watch you as you take a test or a quiz can actually tell whether or not you're cheating, to building neural networks out of dominoes. And it's been a lot of fun to experiment and learn about the different systems that we interact with, some of which I learned about from my research, some of which I never knew about until it happened to come up in, you know, a weird paper or something on social media or something like that. So that's been a great journey for me over the last 2.5 years, and it's something that I definitely plan to keep doing for the rest of my PhD. And then lastly, of course, I have fun. As I mentioned, I'm very into a lot of different sports. So the main ones that I still do are tennis, CrossFit, weightlifting. I still do fence occasionally, but definitely not as much as I did in college. I'm also really into science fiction and fantasy books. So I love reading, mostly fiction right now. And then I used to be into guitar, singing, and travel. Obviously, travel isn't happening right now. And I'm trying to get back into the guitar and singing while I have more time at home. So my dad actually sent me my guitar, which you might be able to see in the background, like 2 months ago, and I still haven't touched it. So hopefully I will get back into that soon. So thank you guys so much for watching this talk, and I'm happy to take any questions.
Thank you so much, Jordan.
Let me stop sharing.
Thanks.
There we go.
Now time for our exciting, absolutely fun Q&As. We got quite a few science-related questions into the chat, into the Q&A pod, Jordan, so I can tell we have a lot of budding scientists listening in. And one of the first questions that we were getting, aside from what's your YouTube channel's name, is what
Oh,
sort of... Oh, go ahead. Why don't you tell us?
No, I was just going to say it's my name. It's just my name.
Jordan Harrod. Okay, so
if you look up Jordan Harrod, you will find it. Yes.
Look her up. Look up these exciting videos. Look at all the cool questions she gets to answer. So, going back to brains for a little bit, the focus of your PhD. You know, we have some scientists, as I mentioned, and you know, scientists love to do experiments. And these students were asking us, a few of them said, you know, Jordan, can you tell us a little bit more about the types of experiments you can do with the brain?
Yeah, so a lot of the experiments that I do right now are focused on deep brain stimulation, which is actually that more invasive method that I mentioned earlier that we're trying to avoid in future versions of these systems, where you essentially do surgery on animals to implant electrodes into their brain. And then based on what you're trying to do, you can implant them in different parts of the brain and then use different frequency and current patterns to try to activate or inactivate different regions of the brain and see what happens.
Got it. So just so I make sure, you know, I mentioned before I am an earth scientist, so I just want to make sure I understand this. So please correct me. But so if I'm understanding this idea of this electrical stimulation of the brain, the reason this works is because the connections and the impulses we make in our brain are dictated by basically electricity, right? It's electrical impulses.
Yes.
So with this deep brain stimulation and the type of work you're trying to do, you're trying to figure out how to fine tune some external electrical sources. So kind of fine tune if we can control how these connections work in the brain so that we don't feel pain. Is that kind of accurate?
Yeah, so general anesthesia it encompasses a lot more things than pain. But pain and consciousness are basically the two things that we're looking to target. And somewhat more specifically, we're looking to target nociception, which is the sensory aspect of pain. So pain is a feeling that your mind essentially creates in order to tell you that something is wrong. Nociception is the sensation of pain that tells your brain to create that feeling. So it's a weird distinction, but when we look to general anesthesia, what we're trying to block in that case is nociception, so that we're definitely sure that you're not feeling pain, because there are also things that nociception does to your body that aren't pain. There are other systems in your body that it affects, and we want to make sure that it also doesn't do that.
Got it. Okay. I mean, I definitely learned something new today. First, I learned the word nociception. So that's the sensory feeling of pain. So that's a cool word, guys. You know, put that on your vocab lists. And this is really cool. So we have a really good question from some of the students who I think might be future electrical engineers. They're asking about these electrodes. So they're asking, so is the goal of your work to use electrodes to eliminate the need for drugs and medicines during surgery?
Ideally, I would say that, so I'm a third-year PhD student. PhDs typically take about 6 years. I would be surprised if we were able to do that over the next 3.5 years. But my goal, I would say, is to get at least one or two of the things that general anesthesia impacts. Hopefully that's pain, hopefully that's consciousness. But yeah, it would be ideal if we could make it so that you don't need any of the drugs that go into general anesthesia. But aside from drugs that affect pain, consciousness, there's also drugs that essentially make sure that your body continues to operate as normal. Because when you knock someone out, it does things to their heart rate and their blood pressure and things like that. And so I'm not sure that we would be able to use a method like this to also control those things. But it would be cool if we could.
Got it. Okay, that's good to know. I mean, one thing that you said during your talk that I connected with. It was an example you were talking about, getting your appendix removed, and you mentioned that story of your colleague actually having your PI Emory as their anesthesiologist. That's incredible. I mean, fun fact for everyone, I don't have an appendix anymore. I had to get my appendix removed back in the day, and I was wondering this is a question about anesthesia, but I honestly don't remember the surgery at all or anything. I was unconscious, presumably. I mean, do we still have the ability to dream during anesthesia? Do you know anything about that?
So anesthesia and sleep are often used as, I guess, analogous things. In reality, they're very different. They're like different brain processes. So you wouldn't be sleeping when you're in surgery. So you probably wouldn't dream. There are cases of, people under anesthesia, either, I don't want to say hearing things because that's also something that can happen, but that usually has more to do with your anesthesia dosage. But experiencing something that is like dreams. But generally, I've been under anesthesia a couple times and I never dreamt. I haven't heard of a ton of people dreaming under anesthesia, so I don't want to say it's not possible, but it's not the same as like sleep.
Got it. Okay, that's a very good distinction to make. So I should not associate sleep and anesthesia so closely. That's good to know, because I definitely don't remember anything, but I consider myself an active dreamer, so that definitely checks out, Jordan.
Yeah.
We have a question about those who are curious about how do you record your experiments when you conduct them? I mean, how do you record them?
Yeah, so it depends somewhat on what kind of experiment it is. Everything gets recorded in some way in my lab notebook, which for me is actually my iPad. I don't use a paper lab notebook because it's just easier to sync things to my laptop and to other computers if I need to. Outside of that, for things like those mouse behavior experiments, we usually have a video recording. So we'll have cameras that are set up in the lab that automatically turn on under certain conditions, or we have in that video a light that automatically turns on when the stimulation is on and turns off when the stimulation is off. We can also record data. So for recording brainwaves, we might just save that data somewhere so that we can analyze it later. For any machine learning stuff that we're doing, we'll save, graphs and accuracy stuff and predictions on different types of data. So everything is saved in some way, usually in some sort of central database. So I have a Dropbox folder where like everything lives. But in terms of specific experiments, it tends to vary based on the experiment and the type of data that you're trying to collect. But everything gets noted in some way in the lab notebook.
Got it. I think I'm very much the same way. I'm still old fashioned, not as high tech as you, Jordan. I'm using a lab notebook that I carry around with me all over the lab when I'm in. I'm actually going to be writing in my lab notebook today after this. So that's definitely good to know. But it's really cool that you have so many different ways and you have the ability to integrate technology into keeping these records. I think that's just so cool. One question we had is, so when animals are in anesthesia, do their brains still function?
Yes. When anyone's under anesthesia, your brain is still functioning on some level. It's just not functioning the same way that it does when you're awake.
Got it. Okay, that's good to know. And one of our perhaps pre-medical students on the call is wondering, what chemicals do they use in anesthesia so that you don't feel pain? So they're asking maybe a contrast from your research with electrodes.
Yeah, so a lot of them are opiates, which is also why anesthesia research and opiate research tends to be somewhat linked. Outside of that, the two, well, I guess there's four that are relatively common: propofol, dex, ketamine more recently, and isoflurane are kind of the big ones. But on top of that, there's also other stuff that's in there to, make sure your blood sugar, or sorry, not your blood sugar, make sure your blood pressure stays stable, make sure that your body temperature stays relatively stable. Generally keep your body at homeostasis, because when you use these drugs, in addition to disrupting pain pathways and sensing pathways, they also disrupt autonomic functions, so things that your body just does without like you having to regulate them. And when that gets disrupted, you need then something to come in and like keep those things going.
Got it. Okay, so what you're saying is that, and these chemicals, can interfere with the processes that are happening in our body automatically. So then we need to kind of jump in and make sure those start running. And sometimes that actually involves additional medicine. So it seems like you could be getting hit by quite a lot during surgery. So that
yeah, general anesthesia is usually several drugs.
Wow. Okay, so you're really making the case as to why your research and working with these electrodes could be really special, because that's kind of reducing the number of medicines involved potentially. That's super cool. So one person was asking something kind of specific about your work, and I'm quite interested in it as well. How do you make sure that the electricity affects only a specific part of the brain? How do you target these regions?
The short answer is modeling. The longer answer is that we have computational models of brains, and you can tell those models, okay, I put electrodes here, 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 and here. I applied this type of current at this type of frequency. Tell me, based on how that field responds to your skull, how it responds to your skin, how it responds to different brain tissues, where things happen, and like if things happen in certain areas and if things don't happen in other areas. So that is, essentially how you decide like where the electrodes go.
Oh my gosh, that's bananas. So you're telling me that computers and the models on our computers are pretty integral to this research. It's not actual physical experiments. A lot of this is happening in these computers through your research.
Yeah, it hasn't always been that way. There were definitely times when there are other ways you can do it. So you can look at like a mouse brain atlas or something and say, I want to hit here. And I know generally that the physics of the system look like this. So I can estimate that I should be placing my electrodes here and here. In human stuff, there's also like weird clinical things where it's like, if you want to hit certain parts of the brain for depression, then like you like put a finger on your face and then like it's like 1 inch next to your finger or something like that. Like it can get a little like wonky in terms of how you actually decide where to stimulate. But for research, we tend to do the modeling.
Got it. Okay, so that's where the computers come in. That's super cool. So Jordan, I mean, systems neuroscientist, you're describing modeling, you're showing us these videos. It seems like you're at your computer quite a bit. Do you get to actually work with these mice directly at all?
Yeah, so I would say that my work is actually probably half animal work and half computation work. It depends a lot on where we are in experiments. So we just wrapped up a bunch of animal work recently, and so now I'm pretty much at my computer all the time. Well, no, we wrapped up a bunch of animal experiments recently, and now I'm 70% at my computer, and then there's device stuff that I'm still doing in lab. So I still go in probably 1 or 2 days a week to work with that. So I would say that, yeah, like 30% of my time is probably spent doing animal work. And then another 20% of my time is spent analyzing data from that animal work. So that's not actually working with the animals, but it is dealing with the data that you collect from those experiments. And then the other half of my time is spent on more machine learning device specific stuff.
Okay, got it. Speaking of your time, I mean, how do you find time to do sports and make amazing YouTube videos and all of this? Can you tell us a little bit about how you found a balance with all of this?
Yeah, so I didn't actually mention all the things that I do in this presentation. So I'm a PhD student. I run a YouTube channel. I run a couple student orgs at MIT, and then try to be active at least once a day. And I think that part of it is just, it's a combination of prioritizing things and then delegating things. So for most weeks, my PhD stuff comes first. I spend, if we look at a given week during like the waking hours, I'm probably spending 75% of my time on PhD things, and that adds up to like a normal 40-hour work week. And then outside of that, things like YouTube tend to happen in the off hours or on the weekends. And it's a little bit easier to fit those things in because a lot of the time I'll be researching something for a PhD related project and I'll happen upon something that would be a cool video. So I'm almost accidentally also doing video research at the same time. And then I can type up scripts or do more research during the off time. And then when it comes to student orgs, the both nice thing and challenging thing about running a lot of student orgs is that if you're at the top, you usually have people underneath you who can manage a lot of the more day-to-day stuff, and you can take a high-level view at whatever the organization is trying to do, which is helpful because it means that for the student orgs that I run, I don't usually have to do a ton like on any specific day. But it does also mean that I end up being in a lot of like high level meetings with other people who are also nerds. And there are times when that kind of gets bumped up the priority list where I know that, you know, we have a conference that we're running on a particular weekend and it's a virtual conference. So I know that I'm going to be on Zoom for like all of Saturday and Sunday. And that just takes precedent.
Got it. Okay, so we need to unfortunately begin our denouement or closing this webinar, but I wanted to ask a question for general advice from you, Jordan. I think that we have a few future systems neuroscientists on the call. Could you briefly tell us, you know, what do you think are the qualities of good neuroscientists?
Yeah, so I think I indirectly addressed this during the talk, but I wanted to make it super clear that one of the things that you definitely don't need is like to have been studying neuroscience for your entire life. So I didn't study any kind of neuroscience until 2.5 years ago. And now I'm doing neuroscience for my PhD. So you can always pick it up. I would say that, like with most scientific fields, a genuine interest in the topic. So if there's, you know, day-to-day aspects of doing neuroscience research that you don't necessarily like, that's fine. Like animal work is not my favorite thing in the world. But there, like you need to either be driven enough by the end goal that you're looking at in order to get through the day-to-day stuff that you don't like, or have a genuine interest in the system that you're looking at on a day-to-day basis. I find that I started my PhD in the first category where I was doing this thing that I thought was really, really cool. And that was what got me to like read the papers and get up to speed on basic neuroscience and like neurobiology. And now it's actually a lot more, I'm genuinely interested in the system, where the more I learn about the brain, the more I'm like, wow, this thing is like so cool and really complicated and we don't really understand a ton about it. And that tends to get me through on a more day-to-day basis. I would also say that, and this is also still a more general, like good traits in grad students, less in neuroscientists, just generally being able to kind of learn on your toes, able to understand your preferred learning style and pick up information in a way that is best for you. Because one of the reasons why I didn't like working in pharma that much was because I found that I had a specific set of skills and those were the only skills that I ever used, and I used them on a bunch of different projects, but I didn't get to learn more about those projects outside of the specific thing that I was being asked to do. And with research, you really get to have ownership over like the whole thing, which I think is really cool, but it also means that there's a lot of stuff that's like not within your wheelhouse that you're going to have to pick up on some level. And some of that is collaboration. Some of that is reaching out to people who know how to do the thing that you're trying to do, because there's certainly stuff that's within my PhD that I don't know how to do, and it doesn't really make sense for me to learn it. It makes a lot more sense for us to just have someone else do it and collaborate with them, and it also makes it more fun. But there's also a lot of stuff that I came in not knowing how to do that I had to learn how to do. And so figuring out how to do that and kind of having that internal drive to keep you going the rest of the way there is probably a big part of it.
That's great. Okay, I think there's a lot of good advice in there for all of us, regardless of which path you take. So I wanted to thank you so much, Jordan, for sharing with us your pathway, your research, your insights. This was absolutely delightful. Before we close, folks, I just wanted to remind folks to please tune in to our next amazing MIT abstract featuring Dr. Sunanda Sharma. She is an interdisciplinary biologist. She helps design how living organisms live and work. And she's also a huge proponent of STEAM, and it's a very integral part of her research. So I'm so excited to have our next talk with Dr. Sunanda. Before we close, I just wanted to also make a big thank you to Nord Anglia for making this series possible. I know I'm certainly really inspired by Jordan, and now brains are on my mind. I'm going to be looking into this for sure after this call. I wanted to thank the MIT-Nord Anglia Collaboration and the fantastic team we have there. And I wanted to also specifically thank the Nord Anglia students who asked us so many excellent questions this time around and during our other abstracts. Folks, I see you're asking all the right questions, and I have no doubt that we have a few future PhDs in the crowd for sure. So thank you so much to everyone for tuning in. Another huge thank you to Jordan Harrod for spending some time with us today. And with that, I'd like to end our webinar, and I'm so excited to see you all the next time around. Thank you so much. Bye, everyone.